At the foundation of every living organism lies chemistry, the study of matter and the interactions that govern its behavior. And in this chapter, we explore how chemical principles shape the structure and function of all living systems. We begin by examining these basic compositions of matter, introducing atoms, elements, and compounds as the building blocks of life, before turning our attention to water and the molecules central to life on Earth, and the unique chemical properties that make biological processes possible. The chapter also highlights the role of essential and trace elements in maintaining life, emphasizing their importance in health and disease, while addressing key questions such as atoms combine to form molecules, why water is vital for life, and how chemical reactions power and sustain living systems. Together, these ideas provide the molecular framework upon which life is built on and sets the foundation for understanding more complex biological systems in the chapters ahead. At the most basic level, biology is chemistry. If you take any biological system apart far enough, you'll eventually reach the chemical level. Matter is defined as anything that occupies space and has mass, which means everything we study in this course qualifies as matter. Living organisms, cells, tissues, and even the air around us forms matter. Mass is simply a measure of how much material an object contains, not how heavy it feels in your hand. All matter is composed of chemical elements. An element is a substance that cannot be broken down into other substances by chemical reactions. Out of the 92 naturally occurring elements, only about 25 are essential for humans. Even more importantly, four elements, oxygen, carbon, hydrogen, and nitrogen, make up about 96% of all the weight of the human body. This tells us that life depends heavily on small numbers of elements. Elements can also combine into form compounds, which are substances that made up of two or more elements in a fixed ratio. The periodic table organizes elements based on their chemical properties and behavior. Each element has a symbol that scientists use as a universal language. While memorization is not the goal, recognizing the periodic table as a tool helps us understand why certain elements interact the way they do. The elements emphasized here are especially important in biological systems and appear repeatedly throughout this course. When we examine the chemical composition of the human body, it becomes clear that the most of our mass comes from just a few elements. Oxygen is the most abundant, making up about 65% of the body, largely because water is such a major component of cells and tissues. Carbon follows at 18.5% and serves as the backbone of all organic molecules, including carbohydrates, lipids, proteins, and nucleic acids. Hydrogens account for about 9.5% and, along with oxygen, contributes to water while also playing a central role in chemical reactions within cells. Nitrogen makes up about 3.3% of the body's mass, which is essential for amino acids and nucleic acids, which means it's critical for proteins and DNA. In a smaller but still important amount, calcium at about 1.5% and phosphorus at about 1% are essential key structures, components of bones and teeth. Other elements such as potassium, sulfur, sodium, chloride, and magnesium appear in fractions of a percentage, yet they are essential for nerve impulses, muscle contractions, fluid balance, and enzyme function. Trace elements together account for less than 0.01% of the body, but are biologically vital. Elements like iron, iodine, zinc, copper, and selenium support oxygen transport, hormone production, and metabolic reaction. Each element is made up of one kind of atom. An atom is the smallest unit of matter that is still retains the properties of an element. Atoms themselves are composed of subatomic particles. Protons carry a positive charge, electrons carry a negative charge, and the neutrons have no charge. Protons and neutrons are located in the nucleus, while electrons move around the nucleus. Even though atoms are incredibly small, their structure determines how elements interact and form the molecules that sustain life. A simplified model of an atom helps visualize how these subatomic particles are arranged. In this example, helium has two protons and two neutrons in its nucleus, with the two electrons moving around the nucleus. The nucleus contains most of the atom's mass, while electrons occupy most of the atom's volume. Although these models are not exact representations, they are useful for understanding atomic structures and how atoms interact with one another. All atoms of a particular element have the same number of protons, and that number is called the atomic number. The atomic number defines the element. The mass number is the sum of the protons and neutrons in the nucleus. Some atoms of the same element can have different numbers of neutrons, and these are called isotopes. Isotopes behave the same in chemical reactions because their number of protons and electrons does not change, but they differ in mass. Some isotopes are radioactive, meaning their nuclei are unstable and decay spontaneously, releasing energy that can be used in medicine and in research. Only electrons are directly involved in chemical reactions. The number and arrangement of electrons in an atom determines its chemical properties. Chemical reactions allow atoms to transfer or share electrons, and these interactions lead to chemical bonds. Chemical bonds are attractions that hold atoms close together. When atoms bond together, they form molecules. Molecules are the building blocks of the substances that make up cells and living organisms. When an atom gains or loses an electron, it becomes electrically charged and is called an ion. Ionic bonds form when oppositely charged ions attract each other. For example, when one atom loses an electron and another one gains it, their opposite charges cause them to stick together. Compounds held together by ionic bonds are called ionic compounds. Table salt is a familiar example. Ionic bonding is common in biological systems, especially in fluids like blood and the cytoplasm, where ions play critical roles in nerve signaling and also muscle contractions. A clear way to understand ionic bonding is to look at what happens between sodium and chlorine. A sodium atom has two electrons in its inner shell, eight electrons in its second shell, and one in its outer third shell. That single outer electron is relatively unstable, and sodium tends to lose it. Chlorine, on the other hand, has two electrons in its inner shell, eight in its second shell, and seven electrons on its outer shell. Chlorine is very close to having a full outer shell, so readily accepts any extra electrons. When sodium gives up its outer electron, it becomes positively charged, sodium ion, written as Na+. Even though it loses an electron, sodium becomes more stable because it now has 8 electrons on its outer shell. Chlorine gains an electron and becomes negatively charged chlorine ion, written as Cl-, which also achieving its outer shell with 8 electrons. The opposite charges of these two ions attract each other, and that attraction is what forms the ionic bond. This strong electrical attraction holds the ions together to form sodium chloride, or NaCl, which is simple table salt. Another major atomic attraction is through covalent bonding. A covalent bond forms when two atoms share one or more pairs of electrons. Instead of one atom taking electrons from another, both atoms benefit by filling their outer electron shell. Covalent bonds are the strongest of the most common chemical bonds, which is why they are also important in living systems. When atoms are held together by a covalent bond, they form a molecule. Many of the molecules essential for life, including sugars, proteins, and DNA, are held together primarily by covalent bonds. Formaldehyde has the molecular formula CH2O, which tells us that the number and type of atoms are present, but not how those atoms are connected. To understand how the molecule is held together, we look at electron configurations and bonding. Carbon sits at the center of the molecule with two hydrogen atoms bonded to it and an oxygen atom positioned above it. Each hydrogen shares a single pair of electrons with carbon, forming two single covalent bonds. The oxygen atom shares two pairs of electrons with carbon, creating a double bond. This sharing of electrons allows each atom to achieve a more stable outer electron shell. Different molecular models help emphasize different aspects of the structure. A structural formula represents each covalent bond as a single line, allowing the double bonds between oxygen and carbon, and a single bond connecting carbon to each hydrogen. A space-field model uses color-coded spheres to show how atoms occupy space, with oxygen shown as red, carbon as black, and hydrogen as white. A ball and stick model also uses color-coded atoms but connects them with sticks, making it easier to see bond angles and overall shapes. Together, these representations show that molecules can be described in multiple ways. Not all bonds involve sharing or transferring electrons completely. Hydrogen bonds are weaker attractions that form when a hydrogen atom covalently bonds to one atom is attracted to another atom with a partial negative charge. Water is a classic example. In a water molecule, oxygen pulls electrons more strongly than hydrogen, creating partial charges. This is uneven sharing of electrons makes water a polar molecule. That polarity allows hydrogen bonds to form between neighboring water molecules. Hydrogen bonding explains many of water's unusual and life supporting properties. Because water molecules are attracted to one another through hydrogen bonds, they tend to stick together. These bonds are individually weak, but together they create a strong collective effect. This bonding is constantly forming and breaking, which allows water to remain liquid under conditions where many other substances would be gas. These interactions are critical for maintaining stable environments inside cells and throughout the ecosystem. Cells are constantly rearranging molecules, and these changes are called chemical reactions. In every chemical reaction, you begin with reactants, which are the starting materials, and end with products, which are the substances that are formed. Chemical reactions can break down bonds, form new ones, or even both. An important principle to remember is that the chemical reactions rearrange matter but do not create or destroy it. The atoms involved are simply reorganized. This constant reshuffling of molecules allows life to grow, maintain itself, and respond to changes in the environment. Life on Earth began in water, and for billions of years, all life existed only in aquatic environments before moving onto land. Even today, modern life remains deeply tied to water. Your cells are composed of about 70 to 95% water, which immediately tells us that water is not just a background substance but a central player in biology. Earth is habitable largely because water is abundant and remains liquid over a wide range of temperatures. The key reason water supports life so well comes down to the polarity in the hydrogen bonds that form between water molecules. Water molecules tend to stick to one another, and this property is called cohesion. Cohesion occurs because hydrogen bonds form between neighboring water molecules, creating a network of attraction. This tendency is much stronger in water than in most other liquids. Cohesion is essential in biological systems, especially in plants, where it helps transport water from the roots all the way up to the leaves. Hydrogen bonding also gives water a high surface tension, meaning the surface of water is relatively difficult to stretch or break. This is why small organisms can sometimes walk on water without sinking. Water resists changes in temperature more than most substances, and hydrogen bonding is again the reason. When water absorbs heat, much of that energy is used to break hydrogen bonds rather than immediately increasing the temperature. As a result, water can absorb a large amount of heat while warming only slightly. When water cools, hydrogen bonds form again, releasing heat into the surrounding environment. This property helps stabilize temperatures in organisms and ecosystems. Evaporative cooling also plays a role, as the evaporation of water removes heat from the surface, helping the organism cool down. When water molecules cool enough to form ice, they arrange themselves into a rigid lattice that spaces the molecules further apart than in liquid water. This structure makes ice less dense than liquid water, which is why ice floats. Floating ice creates an insulating layer on the surface of ponds, lakes, and oceans, reducing heat loss and preventing the water below from freezing completely. As a result, aquatic organisms are able to survive even in extreme cold climates. If ice were more dense than liquid water, it would sink rather than float, causing bodies of water to freeze from the bottom up and making the survival of most aquatic life unlikely. In liquid water, hydrogen bonds are constantly breaking and reforming. Water molecules are always moving, sliding past one another, and briefly forming hydrogen bonds with nearby molecules before those bonds break apart. At any given moment, only some neighboring water molecules are connected by hydrogen bonds. This constant motion is what allows water to remain liquid and flow easily, while still maintaining enough attraction between molecules to support life. When water freezes into ice, the behavior of hydrogen bonds changes dramatically. Stable hydrogen bonds lock the water molecules into a rigid lattice like structure. Each water molecule forms the maximum number of hydrogen bonds, four in total, which forces the molecule to spread further apart, creating the molecules because the molecules are held at a greater distance from one another. Ice is less dense than liquid water. This is why ice floats. That simple physical property has enormous biological consequences because floating ice insulates the water below and allows aquatic life to survive in cold environments. Water is often described as the solvent of life. A solution is a homogeneous mixture of two or more substances. The solvent is the dissolving agent, and the solute is the substance being dissolved. When water is the solvent, the mixture is called an aqueous solution. Water's polarity allows it to dissolve many ionic and polar substances, which makes it ideal for transporting nutrients, gases, and waste products within the organism. Many of the chemical reactions that keep cells alive occur in aqueous solutions. When an ionic compound like sodium chloride is placed in water, the polarity of the water allows the ions to separate and dissolve it. At the edge of the crystals, water molecules surround individual sodium and chloride ions, forming stabilizing shells around them. Water molecules orient themselves differently depending on the charge of the ion. Around a positively charged sodium ion, the oxygen atoms of the water molecules face inward because oxygen has a partial negative charge. Around a negatively charged chloride ion, the hydrogen atoms of the water molecules face inward because hydrogen carries a partial positive charge. These interactions keep the ions separate in solution, explaining why water is such an effective solvent for ionic compounds and why aqueous solutions are important in biological systems. Some substances influence biological systems by changing the concentration of hydrogen ions in the solution. Acids release hydrogen ions or H plus into a solution, increasing acidity. Bases have the opposite effect by accepting hydrogen ions and removing them from solution. Because hydrogen ions are so reactive, even small changes in their concentration can strongly affect cellular processes, including how enzymes function. To describe these changes, scientists use a pH scale which measures hydrogen ion concentration. A low pH indicates a high concentration of hydrogen ions and is more acidic, while high pH reflects a lower concentration and a more basic solution. Each unit changes on the pH scale represents a tenfold change in hydrogen ion concentration, which means relatively small numerical shifts can represent large chemical differences. Most biological systems operate within a very narrow pH range, so maintaining that balance is critical. Buffers help stabilize pH by minimizing changes in hydrogen ion concentration. They work by accepting excess hydrogen ions or donating them when the levels drop. Buffers do not prevent pH changes entirely, but they reduce their magnitude, helping maintain a stable internal environment. This critical stability supports enzyme activity, cellular function, and overall biological balance, reinforcing how closely chemistry and life are connected. Acidity and basicity are determined by a relative concentration of hydrogen and hydroxide ions in a solution, with acidic solutions having higher concentrations of hydrogen ions, neutral solutions having equal concentration of hydrogen and hydroxide ions, and basic solutions having a higher concentration of hydroxide ions. The pH scale ranges from 0 to 14 and provides a way of comparing these differences, with lower values indicating greater acidity and higher values indicating greater basicity. Battery acid falls around pH of 1 to 2, stomach acid and lemon juice near 2, grapefruit juice and soft drinks are around 3, tomato juice and black coffee fall between 4 and 5, and urine, which is slightly acidic, which is about 6. The pure water is neutral at about a pH of 7, within human blood maintained between a pH of 7 and 8. As pH increases, solutions become more basic, with seawater around pH of 8 to 9, milk of magnesia from 10 to 11, household ammonia around 11 to 12, bleach near 12 to 13, and oven cleaner approaching a pH of 14, which highlights how common substances span the scale and why even small pH changes can have actually a significant biological effect.